All sensory systems receive physical stimuli from the environment and transduce the signals to the brain as an electrochemical signal. Different senses have different receptor cells that may be neurons or epithelial cells, and each sense follows a different pathway to the brain. Olfaction, or the sense of smell, transforms chemical odorants into these electrochemical signals that end up in the primary olfactory cortex, or piriform cortex. The olfactory receptor cell is a first-order neuron in the olfactory epithelium. This receptor is a chemoreceptor, as it is activated by chemical energy. Odorants dissolve in the mucosa surrounding the cilia of the receptor cell, which allows them to bind to the receptors on the neuron. The receptors on the cilia are G-protein coupled receptors. When the chemical odorant binds, the G-protein coupled receptor activates a cascade, which results in calcium and sodium ions flowing into the cell, inducing an action potential. Different odorants bind to specific combinations of receptors, resulting in the detection and differentiation of smells. Moving out of the olfactory epithelium, the generated action potentials travel down the axon of the first-order neurons, which synapse with mitral cells, the second-order neurons. These synapses occur at glomeruli located on the olfactory bulb. This is where the specific odorant is differentiated, although not yet perceived, due to the activation of specific combinations of mitral cells. While most other senses have pathways that relay in the thalamus, the olfactory pathway is distinct in that second-order neurons travel straight to the primary olfactory cortex. The mitral cells travel both ipsilateral to the same side of the brain as their cell body, or contralateral, meaning they cross over to the other side of the brain. The olfactory pathway has projections traveling to several other regions of the brain. Some projections travel to the amygdala and hippocampus, areas that are responsible for regulating emotion and memory. This is why smell has strong ties with emotion and memory. Projections also travel to the hypothalamus, which links smell with appetite, as specific neurotransmitters for regulating appetite are produced in the hypothalamus.
In the visual pathway, light travels through the eye to reach the retina, where the visual sensory receptors or photoreceptors are located. There are two types of photoreceptors, rods and cones. Rods are more concentrated in the periphery of the retina. They're sensitive to low levels of light and provide poor acuity, while cones are sensitive to more intense light and provide excellent acuity. Cones are found in the fovea and also provide information about color or hue. Photoreceptors have large segments of membrane that allow greater surface area for photoreceptor proteins, or opsins. In the absence of light, photoreceptors release glutamate neurotransmitters. The second neuron in the pathway, bipolar cells, are inhibitory in the presence of high glutamate concentrations. Specific receptors on bipolar cells cause hyperpolarization when lots of glutamate is present. This prevents any signal from being sent to the brain. When light is present, however, photoreceptors decrease the release of glutamate. This occurs because 11-cis-retinol, a molecule bound to the opsin, absorbs photons of light and changes from its inactive form to all-trans-retinol, its active form. This results in more conformational changes, activating the opsin and G proteins, causing downstream signaling and reduction of glutamate release. Lower concentration of glutamate causes the bipolar cells to depolarize, activating the third neuron in the pathway, a ganglion cell. The active ganglion cell generates the first action potential in the pathway. All three of these neurons are located in the retina. The axon of the ganglion cell projects out of the retina, forming the optic nerve along with the axons of the other ganglion cells. These axons exhibit partial decussation at the optic chiasm, meaning that some of them travel to the same side of the brain as they began, while some cross over the midline to the opposite side. These axons synapse in the thalamus on either side of the brain, at the lateral geniculate nucleus, or LGN. A fourth neuron in the pathway travels from this synapse in the thalamus to the primary visual cortex. Vision is one pathway that has parallel processing. Neurons project from the primary visual cortex to other locations in the brain, each processing a different aspect of vision. The dorsal stream processes the dorsal stream processes information about where objects are and when something is moving, while the ventral stream helps in identifying what an object is. Most of the information in the dorsal stream comes from rods, while the ventral stream gets information mainly from cones.